Uh, Good morning, everyone. And thanks very much for joining the Stormlands TV. And today we are going to look at your life sciences grade 12, and then uh, the topic of nucleic acid. Under the nucleic acid, we're going to outline the following: the location and the structure of both DNA and RNA, the discover of DNA, and then your DNA profiling, your DNA replication, your ribonucleic acid, the difference between DNA and mRNA, and your protein synthesis. Now, nucleic acid. Nucleic acid is an organic compound that contains the element carbon, oxygen, nitrogen, hydrogen, and phosphate. There are two types of nucleic acid, namely your deoxyribonucleic acid, which is your DNA, and your ribonucleic acid, which is your RNA. These two nucleic acids are made up of building blocks, or rather monomers, called the nucleotides. The DNA is found in the form of chromosomes in the nucleus, and these chromosomes carries the heredity information in the form of genes. Now let's look at the structure of DNA itself. It's a double-stranded polynucleotide, also called a double helix. A polynucleotide chain is a very long molecule made up of a string of repeating and similar units called nucleotides. DNA structure. DNA nucleotides consist of three parts as follows: and deoxyribose sugar molecule, phosphate group, and your nitrogen containing base. There are four possible bases that can form part of your DNA nucleotides: one, adenine, thymine, guanine, and your cytosine. Now let's quickly look at the discovery of your DNA. In the early 1950s, Franklin and Morris studied the DNA using the X-ray. These two produced a photograph of the DNA using the X-ray light. On the other hand, there were other two researchers, which namely Watson and Craig. They were also studying the DNA structure, and then Morris conspired with Watson and Craig, and then leaked the X-ray photograph. This allowed Watson and Craig to actually work out the 3D structure of the DNA, and then they later discovered that the structure was a double helix. Now, let's look at DNA profiling. DNA profiling is the process whereby a specific pattern called a profile is obtained from a person or a sample of bodily tissues. In the entire world, every person has their unique DNA, well, except for the identical twins. This means that at some point, DNA can be used to identify an individual, just like a fingerprint. Now, DNA profiling produces patterns of dark bands that are unique to a person. Now, the figure below shows a story where a knife was found at the crime scene, and three suspects were found, and only one of them is guilty. Now, looking at the diagrams here, the one from of the knife from a crime scene, and then suspect number two, the pattern matches the one of the knife from a crime scene, because these dark bands are aligned identically, meaning suspect number two is actually the guilty part here, is actually the guilty part who was in the crime scene. And this is how DNA profiling can be helpful in the forensic department there. Now let's look at the DNA replication. The DNA replication is the process whereby DNA makes an exact copy of itself. The process goes like this. The double helix unwind and the hydrogen bond between nitrogenous bases break and two strands separate, or rather unzip. Each original DNA strand serves as a template upon which the complement is built. The free nucleotides build a DNA strands onto each of the original two DNA strands by attaching to their complementary nitrogenous base. For example, your adenine will bond with your thymine, and your cytosine there with your guanine. Now, this results into identical DNA molecules. Each molecule consists of one original strand and one new strand. The entire process is inspired, or rather influenced, by the enzyme called DNA polymerase. The significance of DNA replication. The importance of DNA replication is to produce two identical copies of DNA molecule, essential for cell division during growth or repair of damaged tissues, and also to ensure that each new cell receives its own copy of the DNA. Now let's look at the ribonucleic acid. RNA molecule is a single-stranded polynucleotide. It is made up of a single chain of nucleotides, and then RNA molecules are shorter than DNA molecules. The nucleotides of RNA differ slightly from those of DNA. An RNA nucleotide consists of a ribose sugar, phosphate group, and one of the four bases. Uracil. Your uracil will replace your thymine, and then you have your cytosine, guanine, and adenine. Uracil is similar to thymine, but it is only found in the RNA molecule replacing the thymine. So now, when you have time, just look at the uracil. The structure of RNA here. You will see your uracil replacing your thymine. Now let's look at the types of RNA. We have messenger RNA, we have ribosomal RNA, and then we have the transfer RNA. Number one, let's look at the messenger here. During the protein synthesis, the messenger RNA carries information about the amino acid sequence of a particular protein from the DNA in the nucleus to the ribosome, where the protein will be formed. Now, secondly, let's look at the ribosomal RNA. Is the RNA component of ribosome a molecular machine that catalyzes the protein synthesis. Its primary function in the protein synthesis is in binding the messenger RNA to ensure that the codon sequence of mRNA is transferred accurately into amino acids in proteins. And then you have your transfer RNA, which carries the amino acids to the ribosomes and matches them to the coded mRNA message. Now let's look at the difference between DNA and RNA. DNA is a double-stranded molecule. RNA is a single-strand molecule. DNA contains deoxyribose. RNA contains ribose sugar. DNA contains the nitrogenous base thymine, and then in case of your RNA you have your uracil, and then DNA molecules are larger, whereas the RNA are shorter. Now let's look at the process of protein synthesis. The process whereby proteins are made. Protein molecules are a long chain of amino acids bonded together by peptide bond. There are two main processes involved in the making of proteins, namely transcription and your translation. Now, looking at your first process there, which is transcription, the diagram here explains everything about the transcription and then also the translation. Now, the end. Thanks very much for watching. Don't forget to subscribe for more, and until we meet again on another topic. Thanks very much.